Namaste students. I am Javin Dr. Rajesh Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic agglutination reaction. This topic belongs to the principles of immunology in the Department of Food and Biotechnology. Students, we take a BT. It is also for the immunology, basic immunology, immunotechnology students also. So agglutination reaction. Agglutination reaction is the type of antigen antibody reaction. In the previous video, we discussed about the precipitation reaction. And agglutination reaction is also a antigen antibody interaction. So let's discuss what is the agglutination reaction. Agglutination is a antigen antibody reaction where the antibody of the serum causes the cellular antigen to adhere to one another to form clumps. So in the agglutination reaction, when an antigen and antibody are in a good combination, then they will create some clumping. Clumps are formed in the antigen. It is the clumping of a particular antigen and its antibody. The antibody that cause agglutination are called the agglutinin or particulate. The particulate antigen include bacteria, viruses, RBC, platelets, lymphocytes, etc. When red blood cells called are agglutinated, the reaction is called the hemagglutination. When the bacterial cells are agglutinated, then it is called the bacterial agglutination. Mechanism. What is the mechanism of the agglutination reaction? Agglutination reaction is brought about by the linking of the antigen and antibodies. And much of the antibodies are bivalent, and antibody can link two antigens. The IgM antibody is multivalent because it is a pentameric structure and it can bind to 5 to 10 combining sites. The IgM antibody had the capacity to make clumps more effectively with lesser number of molecules. The univalent antibodies cannot form clumps because it cannot make some cations due to lack of the binding capability. Agglutination test can be performed to check the concentration of the particular kind of antibody concentration. What are the tests which are used in the agglutination test? And the first one is agglutination ABO, blood group testing, RH blood grouping, Widal test, Vidal test for the typhoid, and the Coombs test for the anti-RH antibodies. So this test is mostly used for the identification of the antibodies. What are the examples? We are going to discuss the examples also. ABO blood group is the first example. In ABO blood grouping, to a uniform suspension of particular antigen, we add a drop of the saline and a drop of the antiserum. In a single test tube, we add 3 drops, 1, 2, 3 drops of the antigen blood. 3 drops, 1, 2, 3. And then we add antiserum, antiserum A, antiserum B, and anti-D. After that, we mix the sample, blood and antiserum are mixed, and we gently observe the clotting here. Okay, it takes few minutes to observe, and it can be observed by the naked eye also. And after few minutes, we observe that there is some clumping like structure. Some clumps are formed in a single drop or two drop or three drop. If there is any kind of clumping, then we can confirm that the blood group is that. And this test is used for the blood grouping and cross matching. Now, tube agglutination. Tube agglutination is also a method to test the agglutination. This is the standard method for the quantitative estimation of antibody. The hemagglutination was used for the testing, qualitative testing. But tube agglutination is a test which is used for the quantitative estimation of antibody. The serum containing the antibody is diluted serially with saline in several small tubes, to which then we add a constant volume of antigen suspension. Constant volume of antigen is added. Then a control tube is kept, which has no serum also, is also added. The tubes are incubated until the visible agglutination is formed. The tube showing the highest agglutination is referred to as the titer, microtiter. The tube agglutination is employed for the serological diagnosis of the typhoid, brucellosis, typhus test, and this test is known as the Vidal test. Here I can share the screen here so that students can understand it easily. So this is the test in which we have added a constant volume of the antigen, and we added the concentration of antibody in a serial dilution. For example, in this tube, the antibody concentration was higher. In this tube, antibody concentration is lower. In this test tube, the concentration is much lower, much lower, much lower, much lower, and much lower. But we added a constant volume of the antigen. After a few minutes, we observe that this tube has no agglutination reaction. This tube has no clumping. This tube do not have any clumping, and this tube also do not have any kind of clumping. So in this tube, we observe some clumping like structure. Uh, Suppose that we have added 1 ml, 2 ml, 3 ml, 4 ml here, 5 ml, 6 ml, 7 ml. So according to this test, we can observe here that the concentration of antibody is 4 ml here. We can find out here what is the concentration of the antibody by using this test, by the serial dilution test. Now, passive agglutination test, it is similar to agglutination test, but the physical nature of the reaction is altered here. In this test, the antigen is coated on the surface of a carrier particle and help to convert the precipitation reaction into agglutination reactions. The carrier particle used can be RBC, latex, sometimes RBC coated with a polystyrene can also be used. This is the passive agglutination. When we are using the passive agglutination is used when there is not any kind of antigen which can bind. So for the binding of the antigen, we are adding a particular carrier molecule so that this antigen can react like a antigen, a real antigen. It is not antigen in nature. So for making it in an antigenic nature, we are adding carrier molecule. After binding to this carrier molecule, the antigen is react as antigen, real antigen. So this coated particle is bound to a antibody and antibody is formed. Okay. Here the epitopes are moved here and carrier molecule is just serving as a body of this antigen and lots of antibodies will bind to these soluble antigen. So what is the reason that we are saying it is a passive agglutination? It is said that the passive agglutination because it is not an antigen, but it's made as an antigen. It is created antigen. So that's why we are calling it as a passive agglutination. So students can be able to understand what is agglutination reaction and what is the suitable experiments to test the agglutination reactions. What are the examples of the agglutination reactions with explanation also. So my lecture is over here. Some questions which can be asked in this, on this topic. That what is the agglutination reactions? Explain agglutination reactions. Explain agglutination reaction with suitable examples. Which test can be used to analyze the agglutination reactions? What is the application of the agglutination reactions? So everything is covered in this video lecture. So my lecture is over here. Thank you very much.